So this is the start of chapter 13's lecture. All right, so I don't know if you guys realize this about Siamese cats. They kind of have a special situation with gene expression. So you guys should know, look at the first bullet, organisms are products of heredity, which means that we're affected by our genes, right? And you should know that. Every gene codes for a protein. These proteins build you and make you who you are. But also their surroundings. We haven't been talking that much about environmental effects. We did a little bit with the cancer stuff, though, right? Okay, so check out Siamese cats. So the dark color, that black colored gene, is only expressed in a cool environment. So go ahead, feel your ears right now and compare that to how your neck temperature is. Big difference? Okay, so what happens is in the warmer environments, the cells that are there on the skin, in the skin for the Siamese cat, if it's warm, the gene is not expressed, so you don't get the dark pigment. So can you see how the Siamese cat then has the dark pigment, the gene being expressed on the extremities, where it's cooler, your fingertips, right? Even that's cooler than that neck. Pretty cool? Okay, so that's your Siamese cat. Then we go on and we talk just a little bit here about identical twins. We will watch a video in the future about this. And identical twins are fun to study because what do they have in common? Okay, they come from the same fertilized egg, so they have the exact same DNA. They have the same genes. So if there is difference between identical twins, we can assume and form a hypothesis and then go test the hypothesis that the differences could be possibly caused by the environment. Now this gets even more interesting when we study separated identical twins that were raised in different environments. Now you might be like, Coop, what in the world? Who's separating twins? That's sick. Okay, so what do you know? What do you know?
There's this pair, so like, because I don't know, they would like give
the one from
like
Africa.
like they should take care of them.
Yeah.
So then there's like these twins. They would separate them and put them into different homes to see if they would act exact same, because like they have the same DNA and everything.
Wait, hold on. There was a real experiment set up to do this?
There's multiple
And like the
And it's
foods
crazy
that they like
No,
are
I
the
don't
same.
know.
It's really creepy.
No, and then it's weird because like there's like this twin boys, the exact same job,
Yep.
almost exact same
The
last
fireman.
name. Yeah, no.
Okay, well, wait, hold on. I've heard these stories before, but you guys have real data, or did we get confused that they were actually separated on purpose for the science, or did we go find separated twins?
There's purpose and accident.
Okay, interesting. I want to research that purpose one. I was unaware that there was. I know about the studies of it, but I might have. Share the link for me on Edmodo. I'll give you a little couple points. Okay, so the deal that I'm aware of, and I'll share it, we got it on video, and it is kind of weird what you guys are talking about, but in times of really hardship, like society, like depression, not people being depressed, during the Great Depression, right? And during like war times and stuff where people they had twins, naturally, right? It wasn't planned or anything like that, and they couldn't, they barely were going to be able to provide for one child. So it wasn't common, but it did happen sometimes where where a mom and dad would decide, or if it was just the mom around, maybe the dad was killed in the war, for example, or something, would give one of the twins to maybe a distant family member or even a close family member, or even friends that couldn't have children even, right? Or had to give one to the orphanage. So these situations happen during hard times, right? So that it's like the Annie story too, right? Where they are trying to give a better life to their offspring, their babies. And then a scientist was trying to find these types of separated twins and get them together and study them, because some of them were even raised in different countries. And so a lot of different environments. So we learn a lot from that sort of stuff. It's pretty interesting. Okay, so then Gregor Mendel is your scientist of genetics. He's called the father of genetics. Look at when he was born, 1822 to 1884, and most of his research was done in 1860s. So a long time ago, and he didn't know nearly what we know now. But one thing that he is so famous for and why he was so successful is he had great scientific design. He designed good experiments that were really controlled, and what he's most famous for is, boy, did he repeat it over and over. He had tons and tons of data, and so that's why his research is so well known, and and it was shared, and people have learned a great things from him. He is a monk, or was a monk, lived in a monastery and took care of the garden. His main thing was the pea, and that's why we have the pea flower model. So let's watch this.
In the 1860s, Gregor Mendel used garden peas to study heredity. Peas are very easy to grow, and Mendel could study many generations during his 8 years of experiments. Peas are also self-fertilizing. The petals of the pea flower completely enclose the reproductive organs. As a result, the pollen from the anthers falls on the stigma of the same flower. Pollen tubes grow down through the female reproductive organ to the ovules in the ovary. The ovules develop into seeds, and the ovary wall develops into the pea pod.
So I've been telling you all year that there's always problems with models, right? So here's our pea model up here. And are the petals large enough? Are they modeling the pea accurately with the large petals? Can these petals cover these parts, the female and male parts? No. But the model chose to misrepresent the petals to make these big so you could see the reproductive organs, right? So it is important that you realize that peas self-fertilize because, I don't care if you know because, but it makes sense, right? The petals keep all the pollen in the same flower. But I want you to know that they self-pollinate because then that allowed Mendel to be using what we call purebreds or true breeding. They were homozygous. They were not hybrids. They were not heterozygous. So he did all these crosses. He crossed his true breds. He crossed his purple flowered peas with some other peas that he came in contact with that were growing in another part of the garden that only had white flowers. And then he studied what he got. He actually got all purple flowered peas. That was weird, what happened to the white. And he kept studying it, so he would do the different generations. Through this, he figured out that the parents had to be passing on something to their babies. So he called it a genetic factor. We didn't know about chromosomes and DNA. And we call it a gene. And you guys know what a gene is, or you better. So does this make sense to you? Segment of DNA whose sequence of nucleotides codes for a specific functional product. What are we talking about most of the time for that product? What do genes code for? All right, come on, you guys. What do genes code for? Proteins. Okay, you've got to know that. Okay, so then this new, well, not necessarily brand new, but vocab word allele. Most of the time there is variation, and you have, at least two different characteristics, like purple or white. Those are known as allele. The purple allele, the white allele. And it's for one particular gene. And he studied lots of different alleles. Purple, white flowers, yellow and green pods, yellow and green peas, round peas versus wrinkled peas, plump or smooth pods versus constricted or wrinkled pods. Kind of reminds me of, snow peas, how the pod is constricted. You could see the pea through the pod. These peas, the dominant is smooth, and they're not like that. Just a little slide here reminding you the difference with chromosomes with bacteria versus eukaryotes. So let's get to our eukaryotes. So our linear chromosomes. So what we have here is we have a photograph of chromosome 7, and we have a picture. Did Nurse Story say that the Noonian, what was it 7? Wait, hold on, that's right. I asked her, she didn't know which chromosome was for that particular disorder. I'm going to look it up. But 21 is Down syndrome, okay? Huh? I think I asked her and she didn't know the chromosome number. So I will find that out, and that might be the one you guys end up choosing for your little mini research paper. I will give you the information on your mini research paper next class. Anyway, the homologs, right? The homologous pairs of chromosomes, they match each other when we use different staining techniques. That makes sense, right? They're the same shape, they have the same genes on them. Might not be an exact copy of the sequence, but extremely similar, so they stain the same way. The newer with the fluorescent on the right here, you can see the two reds, and some of them do stain pretty similar too, but the scientists find more and more techniques, and we could pretty easily pair them up now. Size, location of centromere, and also this old staining technique, which we still use today, that works really well, is this particular stain. On parts of it, it collects more of the stain, so you get these bands. And that's how we can find the homologs. Now, why do we care? Well, we want to look at the person's karyotype. If we think that there could be a chromosomal disorder, we will be able to see it. The other thing, too, is in vitro, when a person is pregnant, we could do an amniocentesis. You've maybe heard of this, where the amniotic fluid is extracted. So the mom gets a little shot to deaden the little area, then they put a bigger shot in, but you can't feel it, right, because they deadened that little spot. And they watch with an ultrasound device to make sure they don't prick the baby, and they pull a little bit of the amniotic fluid out. And the baby's been living in there for a while, and there's baby's skin cells are mixed in the fluid, right, from the baby growing and sloughing off, shedding basically. And we can look at the baby's cells this way. We can find cells or get the cells to go through cell division, and we can get them into prophase, like you saw back here. These have not been manipulated except for stained. So this is in prophase. The nucleus is broken down. The chromosomes have condensed. They're going to start looking for each other if they were going to go through meiosis. If they were going through mitosis, they were just going to line up in metaphase. Because that's probably what they were going to do since we're talking skin cells. Hold on one sec. So anyway, they have this. Well, that's not very organized. So then, and again, they had to shop for a cell that even had the chromosomes kind of spread out anyway. And then they can make a karyotype. You need to remember this vocabulary. A karyotype is an organized picture of the chromosomes for the individual. What were you going to ask? I did not have to get an amniocentesis, thankfully. Just so mom's pretty developed at this time. It depends, actually. It depends if there's been a reason. So, yeah, well, no, that's to the babies. So I don't know if they might do, I don't know where they put the syringe. I would think, okay, so if you're getting amniocentesis, there's a reason to get it. It's a little bit costly, and it's unnecessary unless you think there's a possible chance for some chromosomal disorders. So one, if you're an older mom, for having children, so more than 35, but this is pretty common now, right? There's a higher chance of you having a Down syndrome baby. So they might want to check for amniocentesis. They might want to see if that's the case early on. Now, with any of these things, you guys, you have to realize they're determining if there's anything wrong with the baby. If there's something wrong with the baby, then they're warned, and they're going to know they're going to have a higher risk delivery. Or if it's very early on in pregnancy, they could choose to abort, right? Okay? Now, we're not going to get into if we like that idea or we don't like that idea right now, but, you got to think about the big picture here of what we're doing. Okay, and the mom, and that's up to the mom and dad. So you would only get amniocentesis if there's some sort of high risk for some chromosomal disorders. Maybe it runs in the family, and they're going to do that early on. to do that amnio. So wherever the baby is and how developed it is, they just need to find a little spot, if I understand it straight, correctly, where the baby's not, right? So if you're really full term and they were choosing to do this, and I don't think I guess they would, because they need to, if...
It could be like a disease that doesn't show up until like last minute.
Well, so they do the ultrasounds between typically 18 to 22 weeks. And that is past the 3 months, right, where you're not allowed to get an abortion, unless there was an extreme developmental issue and it was going to be bad for the mom, right, to try and deliver this messed up development of this baby. So it's pretty interesting stuff. Okay, so anyway, so that's all I know about where exactly. I think it just depends on where the baby's at. Yeah, check that out for me. Okay, so there's your karyotype, and you can see they're not lining up perfectly. They're chromosomes, so they're bent, they're smushed, right? They're at different angles. So it is a little bit of work getting them lined up. But this is a normal karyotype. So they went through, they could see the banding pattern is fine, and all these chromosomes are fine. Okay, is this a boy or a girl? So look over here, the 23rd pair, this is an X and an X. The Y is much smaller than an X. But yes, one is kind of smooshed and one's kind of stretched out. Okay? Now, moving on. Yes. Thank you. Okay, so probability. Now, switching gears here. This genetic stuff, using Punnett squares and math, we can figure out the probability. What's the probability that you'll have a boy or you'll have a girl? 50-50. So then we get to things with the alleles like this. That's just the chromosomes, but there's not like dominant and recessive, and there's not genotypes with gender, because we're not looking at individual genes, we're looking at the individual chromosomes. But anyway, here you might have a fourth of a chance that maybe your baby could get a genetic disorder that you know about. And then you could do the genetic screening for these genes. And what about your spouse? What are the odds? So this is very useful. And even deciding if you want to have offspring. Some people might know that something's in their family and they're just going to decide not to chance it. The larger the sample size with any of these probability, the better. So for example, with gambling, I'm sure all of us know people that have lost a lot of money, maybe even homes, even relationships because of gambling. And if you think about it, it's all probably. You're probably going to do what? You're probably going to lose if you choose to gamble. Because the best odds that you could possibly get in a casino are, I think it's right at 30% if you know exactly how to play craps and these particular bets, that's the highest. Most of them are in the high teens and low 20% that you're going to win. So that means, on average, then the casino has about 80% chance they're going to take your money, right? And this leads me to this last part, the larger the sample size. Yes, you hear every once in a while that somebody won a bunch of money, right? So they took a bunch of money from the casino that day. But a lot of other people lost it. And then if you look at the monthly average and then the yearly average, it's very consistent. About 80% of the time you're going to lose. I'm too much of a weenie to be gambling because I feel I work too hard. I don't want to chance that. But it depends on people's personalities. Yes. And still, your odds are still going to be that you're going to... Right. So anyway, so it's the same thing here. Yes, I can throw a coin up and get tails 4 times in a row. Have you guys ever done that? Probably. But it's rare, right, that you're going to get 4 tails in a row. But if I threw that thing 50 times, it's going to be close to 50-50. And it's the same thing with inheritance, with this checking the probability of inheritance and these patterns. Okay, so we want large sample sizes. Okay, a lot of vocab this time. So make sure that you guys are looking for patterns because there's a lot of redundance. So mono, hybrid. What does mono mean? One. Hybrid means it's a heterozygous. It means like a big L and an L. Or a hybrid. Oh, I said that. Heterozygous. Mixed, you could probably even say. And then the P is the parent generation. The F1 is the offspring from the parents. You breed the F1s to make the next generation, and that's the F2. And I'll show you here in a minute. We'll make a Punnett square. Okay, I'm hoping that this is a review. You should know that some traits are dominant, so some alleles are dominant, and some are recessive. And you will always see the one that's dominant even in the presence of the recessive one. Now, most of the time it just makes sense. It's not that the big L is stronger or tougher or whatever it is, like hair color and the petal color of the pea plants. Think about that. If you have a heterozygous, you have a gene for brown hair and you have a gene for the blonde hair. Are you going to see it if the cells are making a blonde pigment and a brown pigment? Are you going to see the blonde color?
Maybe.
It's going to get masked by the darker color. So that's why it's dominant. So from now on, you guys, when you hear something's dominant, think about it and does it make sense? Okay? The epistasis one, I don't think you guys asked me about that for the baby faces. So the chin, that one, the L's, right, that was the L's. If you don't have much of a chin, there's no space for it to be cleft. There's no space for it to have the line down it, right? So they make sense if you think about it, why this one's dominant or why you needed a bigger chin for the other trait to be shown. So think about this. Don't just try and memorize it. They will make sense. Okay, so Mendel. He came up with 3 principles by breeding all these plants, especially his peas. One, he realized that there had to be segregation going on. There had to be a separation between the purple gene and the white gene, because how could the white gene all of a sudden disappear in a generation and pop up the next generation? So he realized, without seeing the chromosomes, without doing the meiosis, knowing about meiosis, he realized that there had to be some separation. And that's called segregation. All right, now there is a little typo on here, so make sure, I know you can barely see it on yours, but if you're looking at the book, make sure you know it's screwed up. The illustrations here are not right. So really quick here, he bred his purebreds. See the big R with the little r, so this is the round seed with the rough seed. Well, that's a purebred. So that one is only going to give away to its gamete, the big R. The other one's only going to give away the little r. If you cross those, you will get all hybrids or heterozygotes. They will all be round. What happened to the rough seed? It's hidden. Okay? Now we breed that generation to make the next generation. We set up the Punnett square. I'm going to wait and show you on another thing. But realize that that should not be wrinkled, that should not be wrinkled, that should not be wrinkled. The ratio should be 3 smooth to 1 wrinkle. That one's screwed up. Now I have a blow up in a second, and we'll look at that. I am pretty confident that you know what these words mean, and if you don't, you need to study. Genotype is just simply standing for the genes, which we symbolize with what? Letters. Capitals and lowercases, that's the genotype. Then we have these words that are either homozygous or heterozygous. Homozygous dominant, homozygous recessive. What's this one called? Heterozygous, because it's different. It has 2 different. The phenotype is what it looks like, and that does depend on dominance. Okay, so here's a Punnett square. So look carefully at that. There are typos. So the big R should not have a wrinkled seed, so maybe take your pen or pencil and go make a nice smooth circle around it. Okay, that should be smooth. Big R, little r should be smooth. Now, how did we make that Punnett square? It's always good to put the cross down. So this is a monohybrid cross. Big R, little r times big R, little r. Draw the Punnett square, and we simply set it up, okay? Let's do one off to the side with different letters. Let's go big L, little l. Okay? So everybody draw a Punnett square off to the side. One of the parents needs to go on one side because we're modeling meiosis. This parent is going to give away what? A big L or a little l. So I just did segregation. I just modeled meiosis. What's going to be over on this side of the Punnett square? Big L, little l. Put them together. Capitals always come first. Questions on putting that together? Okay, so for both of these scenarios, we got the same results. So then there's the genotypic ratio. So the genotypic ratio is what genotypes we have. We have one of big one, big one. We have two of hetero, and we have one of fat one. So the genotypic ratio is a 1 to 2 to 1. We go the dominant, most dominant, right to the recessive order. That's your genotypic ratio. I write that down. Phenotypic ratio is what? Just like up here on the P's. We have the dominant trait. That should be smooth. How many smooth seeds should we have? 3. So it's 3 to... Notice how they add up to the number of boxes in your Punnett squares. These are not fractions, they're ratios. You could say we had 2 heteros out of 4, but that's not a ratio, that's a fraction. Okay? And you could also figure out percentage. These are your predictions. Okay, then we get to the cool one, the dihybrid cross. So, with the dihybrid cross, I got it written right here. Let's work with Y's and R's because that's what you're going to work with later in your lab. So, yellow is dominant over green, and it's the same thing like we just did, round seed versus wrinkle seed. So dihybrid because di means 2, and they're all hybrids, right? Now, you're going to make a big Punnett square. Wait, before I move and show it to you. When he did these crosses, he realized that these traits independently assort. It wasn't like you had more odds of having a pink flower with a green seed. It was just random. And that's different than some of the traits that are linked. For example, you guys know that it's more often that people that have blonde hair have light eyes, like blue or green eyes, than brown. Okay, it still can be mixed up, and we'll talk more about that. Crossing over mixes that up. But those genes are found on the same chromosome, so they have less odds of getting mixed up because they're inherited together commonly. But not always, because crossing over could separate them. But all the traits Mendel were looking at were on different chromosomes, so they independently assorted. And he learned this by doing this large Punnett square. You guys are doing great, by the way. Hang in there with me. We're almost done. With 16 boxes. Now, the setup is what I think is hard, figuring out the gametes. So look at our genotype. You can only have one of those in the gamete, and you can only have one of the R's in the gamete. You guys with me? So here, a little math. So we use FOIL to figure out all the possible combination of gametes. You guys join me? So big Y and big R are one of the possibilities, correct? See it over here on the Punnett square? Okay, then big Y and little r are a possibility. Little y and big R, and the little one. Notice that we want to keep our genes together when we do this, so stay in the same order. So see how then both sides will be set up the same because the parents are the same. right? And then look how you put them together. You keep your R's together. Since they're going first, keep them going first. And then you put your Y's together. Most of the time I write it out. I don't put it on different lines like they kind of did, but it doesn't matter. The other big thing to make sure you realize is make your Y's or your P's or whatever, make your capitals very different from your lowercase. You'll get a headache trying to look at it if you can't remember what you were drawing. So I never draw little P's with that little thing like that, but I do in genetics, so I don't get confused on my P's. My Y's, the same thing, very different looking, so I don't screw that up. Okay, now, doing a genotypic ratio for 16 like that is too complicated. We don't care. We care more about the phenotypes. We can do a phenotypic ratio. They color-coded it for you, but you should be able to look at the genotypes and determine what it is. Big R is going to be smooth, the round seed. And let's go to this one over here. Little y is going to be green, okay? So, but the color coding is nice. How many round yellows do we have? 2, 3, 4, 5, 6, 7, 8. How many? 9. Now let's go in order. Let's stay with the round. So how many round greens? Round greens together. How many yellow wrinkled? How many recessive? 1. That is the genotypic ratio for a dihybrid cross. 2 more slides and we're done for today. A little bit more with that sex determination. I just want you to realize that yes, humans have the XY setup. Even fruit flies have that, and mammals. So it's a pretty common system of genders. But hold on, I'll get to some other ones.
The chromosomes of human patients are XY in a male and XX in a female. The result after 2 meiotic divisions is sperm with either an X or a Y chromosome and an egg with an X chromosome. Sex in humans is determined by the sex chromosomes of the egg and the sperm at the moment of fertilization. XX produces a female. XY produces a male.
Okay, so that sounded familiar, didn't it? Okay, so insects, a lot of them, not all of them, but like I already mentioned bees to you guys, and even that crazy lizard was like this, that whiptail lizard in your book. The females are diploid, have the 2 X's. The males are actually haploid. They don't have a Y, they just have 1 X, and that makes them a male. But then there are things, like I heard somebody say something about fish, some fish and birds, and some insects actually have it reversed, where the male has the matching chromosomes, like the X and the X, and the female have the X and the Y. And then they decided to give them different letters to represent that. So then the males are ZZ, and the female is a ZW. And then there are some things that are hermaphrodites, like our peas and lots of our plants. They have both male and female parts in one, right? So there are no genders. They're capable of self-fertilization, or they can send their pollen off and still have sexual reproduction. And so there are no different chromosomes, no sex chromosomes for that. Okay, you survived. We're going to stop there.